Hey guys, welcome back to our new video. So in this session that we are going to discuss about the topic enzymes. So enzymes are a biocatalyst as you all know. A catalyst is defined as a substance that increases the velocity or the rate of a chemical reaction without itself undergoing any change in the overall process. So enzymes can be defined as a biocatalyst synthesized by living cell, and normally they are protein in nature, colloidal and thermolabile in character, and specific in their action. So this was a very short definition about the term enzyme. So next let's dig into a bit more detail. So let's discuss on the classification of enzymes. So the enzymes are sometimes considered under major two categories. The very first one is intracellular enzyme. So as the term suggests that. So intracellular enzymes means they function within the cell where they are synthesized. And the second category is extracellular enzymes. So extracellular means which is external to the cell or outside the cell. So these are the enzymes that are acting outside the cell. And apart from this small classification, the enzymes are divided into six major classes, and each classes on its own represents a general type of reaction about by the enzymes of that classes. So let's talk about these six major classes. So the very first class is oxidoreductase. So these are the enzymes that catalyzes oxidation and reduction reactions. For example, pyruvate dehydrogenase, which catalyzes the oxidation of pyruvate to acetyl CoA. And the second example is alcohol dehydrogenase. And the third example is glucose 6 phosphate dehydrogenase. And the fourth example is isocitrate dehydrogenase. And coming to the second class, which is transferase. So these enzymes catalyzes the transfer of a chemical group from one compound to another. For example, hexokinase, which transfer the hexose to hexose 6 phosphate. And coming to the third classes, hydrolases. So these enzymes catalyze the hydrolysis of a chemical compound. For example, the enzyme pepsin hydrolases peptide bond in protein. Coming to the third classes, lyases. So these are the enzymes specialized in the addition or the removal of ammonia and carbon dioxide. For example, decarboxylase have an action on the tryptophan to tryptamine plus carbon dioxide. And apart from that, the enzyme isocitrate dehydrogenase have an effect on oxalosuccinate to alpha ketoglutarate. And coming to the fifth classes, isomerase. So these are the classes of enzymes that can catalyze the formation of an isomer of a compound. For example, the enzyme phosphoglucomutase, which can catalyze the formation of glucose 6 phosphate from glucose 1 phosphate. And apart from that, the another example is the enzyme glucose 6 phosphate isomerase will act on glucose 6 phosphate to form fructose 6 phosphate. And coming to our last classes, ligases. So these are the enzymes which catalyzing the synthetic reaction when two molecules are joined together and for which the ATP is used. For example, the DNA ligase catalyzes the joining of two fragments of DNA by forming a phosphodiester bond. Or the other example is in urea cycle, carbon dioxide plus ammonia results in the formation of carbamyl phosphate by the presence of the enzyme carbamyl phosphate synthase. So these are the six major classes of enzyme: oxidoreductase, transferase, hydrolases, lyases, isomerase, and ligases. So this is all about today's video, and in next video we will be talking about some of the major factors which are affecting the enzyme activity. So thanks for your watching. Please do subscribe to my channel. See you soon.